I know everyone's here not to hear about events that are coming up, but to be at this event and to chat with Professor Church. And it gives me extreme delight to introduce him. I'm sure you guys know all the accolades. Faculty member at HMS, founder, one of the co-founders of the Wyss Institute. One of the, in my opinion, one of the Edisons of our time, one of the most noted and celebrated scientists alive today. When I chatted with Professor Church earlier this year, very, very casually, he mentioned that his lab had churned out more than 12 startups last year. And this is a pace that's very much unheard of for most other labs. Not only is he a serial entrepreneur, but he is a mentor who empowers the students in his lab to start companies that change the world. And so it gives me absolute pleasure to introduce Professor Church. He'll chat for 5 to 10 minutes on kind of whatever he feels free to do. And then there were 57 questions that were pre-submitted, the most questions I've ever had as a moderator. So I know everyone here is extremely excited. And we can ask those, or we can ask stuff kind of in chat. So I'd welcome you guys to start asking your questions in chat. I'll help curate that. But with that, I'm going to get out of the way apart from that and just turn it over to Professor Church.
Thank you very much. That's a great intro. And I'm really just looking forward to a Q&A session. I'll just show a couple of slides just to show that I can show slides. But think of it more as a kaleidoscope than as an exposition. And if it looks technical, you can ask any question at any level. Don't worry about the slides. This is by conflict of interest. Used to fit on a slide. Now it's on a web page. It's complicated. But we're going to talk about the future of synthetic biology. We have a number of different approaches to SARS-CoV-2. Obviously, on everybody's mind, we're working on treatments, neutralizing antibodies, peptides, and so forth. We have a radical vaccine that I'm sure will come up in discussion. It's one of the very, very few that's intranasally delivered, maybe 5 out of 207. It's got peptides, it's got everything having to do with chitosan nanoparticles. And then bioweather map is something that's a little bit forward-looking. It handles SARS, but it also handles all the next set of pathogens. So keep that buzzword in mind, bioweather map. We've been aiming at it since 2002, but I think we're finally going to get it. We're woefully underdiagnosed. We can't tell the difference between things that are completely harmless and things that are deadly. Again, bioweather maps, I think, is something we should have proactively. And part of the reason we can even have this conversation of bio as if it were the same cost as weather maps is that it has come down in price 60 million fold in just a few years. Our group and others that we collaborate with have gone through a whole series of generations of so-called next generation sequencing and next generation synthesis, both synthesis and reading and writing. So we brought it down from a $3 billion poor genome to a $100 clinical grade genome. We're trying to do the same thing for therapies, providing low-cost alternatives. There are 3 or 4 major categories of approaches: rare genetic diseases, infectious diseases, or prevention. Common diseases we're categorizing as age-affected, so we're working on aging reversal. And then a 4th is machine learning for making a higher success rate. We have imaging methods, imaging now that can go down to 20 nanometer resolution, the order of molecular scale, all the way up to brain-sized imaging. 2 different companies involved here, ReadCore and Bruker. So we can see little tiny loops in the genome. We can see RNA, DNA, RNA, and protein. Here are the synapses at super resolution. And then we can use machine learning. We use it on antibodies, on using protein barcodes to allow us to do multiple, essentially clinical or preclinical trials simultaneously in single organisms. We can do thousands to millions of different tests at once. So, for example, we've put 1.2 million different capsid designs for gene therapy delivery into one animal. We can do this multiple times, and then we can use machine learning loops to get better and better at delivery to certain tissues or to or different immune systems. We can also do, so that's at the sort of protein and viral deliveries level. We can also do it at the cellular level, where we can do multiplex analysis of different recipes for making different cell types, making supercells or cell therapies, or transplantable organs. So GC Therapeutics is working on these supercells that, for example, not only can replace cells that are destroyed by demyelinating diseases, such as this one in an animal model, but can replace them in a way that they themselves are not susceptible to the demyelinating disease. And here we've restored the functionality by injecting these human derived organs into mouse brains. That's GC Therapeutics. Then we can make even more complicated organs, the full set that are transplantable between humans, in a way that doesn't require live or dead human donors. But this requires a very exotic and comprehensive germline editing of pigs. So 42 different edits to the sugars, clotting factors, immune components, and getting rid of their endogenous retroviruses. So for the first time in history, we've gotten rid of all of the endogenous retroviruses from a mammal. This is in pigs. And these are in preclinical primate trials at Massachusetts General Hospital under the direction of Jim Markman. And this has all been spearheaded by Luhan Yang, who was one of the co-inventors of CRISPR when she was in my lab, and has co-founded Kihan and Regenesis in 2 different countries. And then finally, we're trying to make flight recorders for human body, or bodies in general, plants, animals, and humans. This is like a flight recorder. You record all kinds of information from all different parts of the plane, and then you only look at it if something bad goes down, and you look at parts of it. And we do this with a variety of molecular tools, including CRISPR, but not limited to it, where we've adapted it so that it acts as a recording device, not just of the developmental biology, where every cell records its personal history, but you can also record things like hypoxia or essentially any molecular event. And this is, again, Prashant Mali was one of the co-inventors of CRISPR, and he worked with Reja Kalar. We can make cells resistant to many or all viruses. We think we can make any organism resistant, microbes, plants, animals, and human cells. We do this not by getting rid of individual viral receptors, some of which are required for the organism to survive, but by changing the genetic code, the triplet code, where there's 64 triplets. And we've proven this for one organism. We've completely recoded the entire, all of the examples of this codon genome-wide over in these 3 Science and Nature papers, and have shown that it is not just a little bit resistant, but completely resistant to 92% of the viral types, so that you can put in up to 1 trillion viruses per milliliter and get 0 viruses out. And I just thank my lab. This is our lab meeting where we're kind of showing off our masks, and that was your kaleidoscope. But now let's get to the questions. That's and they're not limited to what I just talked about. Any topic that you know of, and any that come up while we're talking. Thank you.
Great, thank you, Professor Church. I'm going to start with the softball. The first question that I see that could be very widely construed is, I suspect, from a student or a graduate student, specifically asking, what skills do you most value in the young people who join your labs?
Certainly open-mindedness. You know, it appears that we take risks, but really we just have things that look hard to other groups, things that look useless or expensive, we can see that they're not intrinsically so. There's no law of physics, and so we're looking for things where we can quickly quickly test that. The other thing is so that kind of experimental willingness is one, and being nice. People are sometimes surprised that my top interview question is not about genius; it's about being nice.
Awesome. We're going to continue with softballs just for a bit, and then we're going to get into some of the complicated questions that folks are asking. Sure. So I like this question next, and it reminds me of a conversation I had with the CEO of HubSpot, who mentioned that you're the smartest person in Boston. You have a reputation for being a genius. And so the question is, what does Professor Church find difficult?
Well, I find it difficult to live up to that particular reputation, for one thing. Boston is an extremely smart city, and we're an extremely smart world. But I think we have probably a little bit more trouble publishing and raising funds than you would expect. But fortunately, we're very frugal and very patient. So the little bit of money we have goes a long way, and certainly I can't complain. And delays in publishing, we compensate by publishing preprints and just trying to answer the referees. But yeah, to give us some idea of the funding situation, we estimate that two of our favorite technologies that we played a big role in and took them all the way from a crazy idea to something that was completely commercially viable, was next-gen sequencing and editing, and we estimate the cost our lab about $2 million each. So the return on investment there is easily billions for each of them. We weren't the only one in either of those fields, but could have been.
Incredible. Last easy question, and we're going to move away from the softballs after it. This is another student, I think. What advice would you give to young students interested in brain science today?
Well, almost any science, I would say, add the term synthetic biology to it, and in some cases add the term imaging or super resolution imaging. Certainly for brain, those two are quite relevant. You want to be able to examine every aspect of the brain, its developmental biology, its synapses, synaptic connectome, etc., at any resolution down to at least 20 nanometers. And then you'd like to not just read it, but write it. And that's what the synthetic biology is about. You like to build brains to test ideas and to make new products.
Awesome. Let's start digging in. And we're now moving kind of into the question of future of SynBio, regulatory questions, etc. First one on my mind is, what is your guess about the areas in this domain that will have breakthroughs first, and how will this make a difference in what happens next?
Well, I think some of the fastest breakthroughs are the ones that don't require FDA approval. I mean, I'm a big fan of FDA. We shouldn't go any faster for things that really have a risk profile where you need to spend 10 years. You can see a little bit of acceleration when it comes to COVID-19 because the benefits greatly outweigh the risks, especially for non-risky vaccines. So some of the first things will be diagnostics. A lot of preventative medicine is, I think, underappreciated. We could have avoided $10 trillion of COVID-related costs, and agonizing excess million deaths by having better readouts. It's very cost effective now. It's come down by 60 million fold. There's really no excuse for us not to be doing that sort of thing. And so that'll be the first. And a lot of that does not require, it benefits from, but does not require FDA approval. Some of it will be formal diagnostics, some of it will be theragnostics, but some of it will be do-it-yourself or home tests that are less formal.
Some questions came in through chat that are clarificatory questions, so we'll get to them before we get to future of questions. Sid Mehta asked, if one edits genes, can this cause unforeseeable problems a few years down the line or a few generations down? How do you measure this risk?
You certainly should think about a few generations down for all new technologies. Do cell phones cause risks a few generations down? Not by mutating your brain, but by changing our social fabric. Do conventional therapeutics have affect future generations? We know there's epigenetic effects that happen, for example, after starvation in the world wars. That said, it's really about whether the benefits outweigh the risks, and you can't wait for a multi-generation clinical trial to declare that we're now in phase 4. And then in phase 4, we have a chance of picking up those things. We always are using theory plus clinical trials to hopefully prevent those longer term effects. Most of the failures that we've seen are things like thalidomide, hormone replacement therapy, Vioxx. These have been caught pretty quickly in phase 4.
Cool, thank you. And then similarly, Stefan Lukyanov also had an on-the-spot question. He asks, Dr. Church, do any of the companies derived from your lab focus on virus-based therapies for diseases in relatively inaccessible tissues such as brain, connective, etc.? Thank you.
Yes, so we have 3 companies that are working one way or another with viral delivery of gene therapies. Dino is using the machine learning that I mentioned to evolve more efficient delivery and more targeted, so both systemically and in a very tissue and cell type specific manner. Ally is working on ways to finesse the innate immune system, and Regenesis Bio is using gene therapies to deliver aging reversal genes. In every case, I think at this point, gene delivery is the big promising opportunity right now.
That makes a lot of sense. We're getting back to broad future of SynBio questions, and I'm going to lump 2 together because I think one almost speaks to part of the answer for the first. So the question is, what social cultural roadblocks will be the most important in the maturation of synthetic biology? And then a sub-question to that is, when, if ever, do you think will Europe allow genome-edited plants to be regulated in the manner of mutation breeding plants rather than the current regulatory path for GMOs?
So yeah, you're right. They are closely related, and I think answering the second one will do it all. I think that Europe is open to all kinds of GMOs already, including bacteria that make human proteins, human insulin, say, also to humans, so that are walking around with gene therapies. Are a kind of GMO. But more significantly, we're in there's a big decision that has to be, that Europeans in particular have to make, which is if you prevent a simple mutation like an A to a G from being allowed, if you only allow it if it happened in nature randomly, or in a conventional farm mutagenesis protocol where you might expose it to sunlight, ultraviolet radiation or something, then you will have lots of random mutations rather than one precise one. And if you find something in the wild, you'll always have a question, if someone claims it came from the wild, did it really come from the wild or did they plant it there? And so I think that it's going to be very difficult, very challenging to regulate single point mutations. It was much easier to regulate transgenics where you move an entire cluster of bacterial genes into a plant. That's easy to detect with PCR and other methods. I think that detecting a single base, one out of billions of bases that could occur at random is both unhelpful and it reduces your confidence in being able to use naturally occurring variation.
Super. This might be an interesting follow-up question to that, which is, if we were to reconvene 5 years from now, what is your best guess as to what new developments in the field will have taken place?
Well, there's a lot of the field here. If it's all of synthetic biology, that would take up all the rest of our time and still have open ends. But I think the accelerated developmental biology is, many of these things are not even limited to biology and medicine. So accelerated developmental biology might be a better way of doing 3D printing. So rather than 3D printing helping developmental biology, it could be the other way around. In developmental biology, you have like trillions of print heads per cubic millimeter. And that has implications for making new kinds of computing, which are actual neural networks rather than silicon simulated ones, and new ways of doing quantum computing as well, where you're actually making complex chemical and physical systems, which are again better than the simulations, faster, cheaper, more accurate than the simulations. So simulations might be replaced by the real thing, in this case when they're extremely inexpensive to make billions of trillions of experiments at once.
Cool. I'm noticing as I go through the questions, a lot of questions are about time. Like, when is this going to happen? When is this going to happen? And so another one kind of in line for that in the context of the future is, will we soon see on-demand produced cheap DNA of great length? And why is long DNA not yet available?
Right. So you can synthesize a human genome very easily. It's thousands of dollars. It's just not long. That's what the question is about. It's about assembly rather than synthesis. We may change that. So if we can get very accurate, very long synthesis, say enzymatic rather than organic chemistry has trouble making things bigger than 300 base pairs long, while enzymatics routinely makes things that are hundreds of millions of base pairs long in a replication sense. But having it instructed from a digital computer file is a challenging thing, and to get that to be accurate. The assembly problem has largely to do with repeats and other repetitive DNA and other DNA structures that get hung up when they're single-stranded or otherwise recombining. I think we're on an exponential curve, and for almost all these technologies, and DNA assembly is one of them. So we've launched a project called Genome Project Write that aims to be able to write any length DNA in any organism, and I think that will improve exponentially. So all these questions about timing, which you said, the answer is it takes 10 years to do a typical clinical trial. So that's one kind of timing that is kind of a given for most of these things. And then if it doesn't involve a clinical trial, then we're on an exponential curve where we're doubling speed, cost, and accuracy about once a year or faster than that.
I love it. As I'm realizing, the questions are not just about speed, they're also focusing on gene therapies, curing diseases, and then everything else that SynBio can kind of touch on. So I'm, for the purposes of this discussion, I'm just going to go through the medical questions first, then we'll move on to kind of everything else to try to keep everything organized. With that in mind, a question I see is, when will synthetic biology cure a disease like Lyme or malaria?
Um, well, so it's already curing disease. Gene therapies are already approved by FDA for general use. So that's curing quite a few generally rare diseases. Um, I think we'll see more common diseases that are treated as diseases of aging. Many diseases, almost every morbidity and mortality is increased with age. So our COVID-19 goes up steeply with age. Falling down and not getting up goes up with age and so forth. I think all of those will be are on a fast track for gene therapy and other solutions. There was another part to the question.
You answered it directly. I think that's fine.
Okay.
What it is specifically about when a disease like Lyme or malaria will be cured, but I think it was
Lyme and malaria. Those are hard. And diseases that affect poorer populations like malaria, HIV, TB, guinea worm, so forth. Those have been challenging simply because the profit motive is not as strong. Lyme does affect wealthy individuals, especially in the Northeast of America, but it is a global disease now. There was a nice preventative for Lyme, which was a vaccine, which worked. In fact, it works so well that dogs are prevented from getting Lyme disease by vaccines. So there are now 2 companies that are bringing new vaccines, which are very similar to the old vaccine, onto the market for clinical trials. And so I'm hoping that healthy competition and revitalization will be a big part of it. And we're also, our lab has funding to also look into some of the later Lyme disease-related symptoms that were initially neglected and dismissed. But I think that's an important part of it, and that involves looking at the immune system tolerance. And so tolerance is something that's important not just for Lyme disease, but for multiple autoimmune diseases and transplants. And so most of the technology we develop have multiple uses. So sequencing can be used for all kinds of things, synthetic biology for a lot of things, and in this case, immune tolerance has a lot of applications, including Lyme disease.
interesting. Kind of in that same line, how do you see the future of human microbiome manipulation to improve health outcomes, especially for those suffering from autoimmune conditions?
Well, I think this is something where we can get, we might avoid over-promising and hopefully over-delivering later. I think it's it's beguiling to hope that there's something that you eat or don't eat, or drink or don't drink, is enough to give you long life or better health. I think we need to be cautious with microbiomics, but there's hard science showing that there is exchange of metabolites between the gut and the blood and the brain. All of those are in equilibrium. And so in principle, it is yet another way of delivering enzymes that can detoxify and so forth. Certainly dysfunction of your microbiome is proven scientifically as well. Clostridium difficile is the poster child for that. So I think the time has come for it. And there'll also be various conveniences where the microbiome causes you various personal and social distress. So that it's a great field.
Super interesting. changing gears, but not too much, not too far away. We've got a question for, do you regard SynBio as holding promise for curing neurodevelopmental disabilities such as ADHD or autism? If so, in your judgment, which disorders and how far in the future are such cures? Always how far in the future, always timeline.
Well, some of them are literally in the past if we just adopted them. So that's a direct answer to when. So many neurodevelopmental disorders, thousands of them, have a genetic component which is understood and can be prevented by genetic counseling. And we're trying to bring that. We brought the cost down of the diagnosis down in the sort of $100 range, and now we're trying to, get people used to that idea in various ways. So people don't routinely go to genetic counselors unless they have a family history, but most people that have a genetic disease do not have a family history. So we need to get people used to doing it preventatively, just like seatbelts and, annual medical checkups. And so one of the ways we're doing that is via a dating service, because a lot of the decision making that can lead to genetic diseases is occurring around that time in your life or that event. So that's the shortest, that's the fastest. Meaning it's the solution exists. We just need to get people to use it, and it's dirt cheap and very accurate. For people that are already born, which is most of us, 7.7 billion of this, we're getting much better at gene delivery to the brain. It's crossing the blood-brain barrier is something that some of our white blood cells, leukocytes, do all the time, and so we're working on using those as delivery vehicles. That will take longer. That will take at least a decade to get through the clinical trials, probably 2.
Awesome. I want us to start slowly shifting over to the broader applications of SynBio. Before we get to it, there are some questions that are not quite hardballs and maybe ask for a little bit more explanation about semantics. And so I think it could be valuable to ask those as well. Specifically, I'm looking at a question that says, is there a distinction between synthetic biology and regenerative medicine? And I think it's a good opportunity to kind of speak about the semantics in the field.
Yeah, so regenerative medicine, I think, is a subset of synthetic biology. Synthetic biology, some people want to restrict it to molecular biology or even restrict it even more to like molecular circuits. I think that's unnecessary. Anything that you think of is not found in the wild is probably, but is still biological or biomedical, is probably synthetic biology. So regenerative medicine, I think, is going to get a boost from our ability we can now reprogram almost any cell into any other cell type. We can make those into organs, either in cell culture or in animals like pigs. And so that opens up whole new possibilities for regenerative medicine. We have essentially enhanced cells, enhanced organs that are immune tolerant, virus or pathogen resistant, senescence resistance, and cancer resistant. All of those have been shown in animals. Now it's just a matter of translating them into a form of cell or organ that we can get into patients.
Thank you. So the promise of synthetic biology is to edit cells to do useful things at scale. And that at scale question has kind of been a question mark in many spaces for SynBio. Mariana Mila-Husova in chat asks, what do we need? How far away are we from directed enzyme evolution plus machine learning that could lead to enzymes for any chemical reaction at commercializable scale?
Well, I'm going to break that in half. I mean, scaling is actually the easy part. If you have something that works well in the laboratory, getting it to work in the farm or the fermenter or the patient isn't trivial, but at least it's kind of it's well exercised engineering, where we know where we know how where the pitfalls are, how to get through the trials. So the discovery of an enzyme that will do any chemical reaction, that can range from really easy. So if it's related to a chemical reaction that already occurs via enzymes, then that's easy. And then at the hard end are things where it just seems like the temperatures involved. Sometimes you can bring the temperature down from 800 degrees to room temperature, but other times it's less obvious. So I would use as examples, carbon allotropes, carbon nanotubes, buckyballs, and diamonoids are going to be challenging to make enzymes for those, but useful if we can figure it out. But the sort of things that involve biomolecules, the combination of big libraries, meaning millions of designed enzymes, and machine learning is something that we routinely do in our lab, and we published a handful of papers recently that just leapfrogs over all the computation that was limited to molecular dynamics or other simulations of proteins. Now we just use combinations of big libraries of information where we know how proteins assemble, plus big libraries of design proteins to see how they behave.
Lovely. We're shifting now. And when we think about questions for SynBio in non-medical applications, the questions that keep popping up are in the realm of food and the food industry. So I'm going to read a series of those questions because they're all interrelated, and perhaps you can kind of bunch them up in your response. So the questions are, how is synthetic biology going to change the foods industry in general and the fruit industry in particular? Second, could you speak to the use of precision fermentation technology in food applications, specifically in dairy or cheese alternatives? And then finally, what is the role of SynBio in food and agriculture innovation?
Right. So agriculture is tough. It's like right up there with biofuels in the sense that it's highly commoditized and low cost, and low profit margins. So you have to create something that's either exotic, or otherwise unusual. Nutrition is relatively a solved problem, at least for wealthy individuals, and that's why the wealthy nations have been the most intolerant when it comes to GMOs, because they have the least to gain. Less wealthy benefit from something even as simple as providing vitamin A in golden rice. And that's now getting adopted in some of those poor countries like the Philippines. I think in general, vegetable matter is more cost effective, both in terms of water use, less antibiotic use, less zoonotic diseases, etc. So synthetic biology will work on that. There are synthetic biology arising organisms that appear to have advantages. So, for example, some that have doubling times of less than 3 hours, which compares very favorably to vascular plants, where the doubling time is measured in months. What was the last part of the question? I think there was one more part to it.
There were questions on dairy, on
dairy,
fruits.
Okay. So dairy, a lot of dairy products have a viral problem. It's no coincidence that one of the first applications of CRISPR was in dairy products like yogurt. And we have now solutions where we can make them completely virus resistant. CRISPR really is not up to the task. It helps in the wild, it helps in the industry, but it's really, we can make them completely virus resistant by engineering the genetic code. So I think that will probably happen in the dairy industry. We can also replace the cow part of the dairy industry and provide all the proteins that the cow provides from a completely vegetable or even microbial source, like these rapidly doubling cyanobacteria. So getting everything up to the point that, and the source of energy can either be from the sun, which is classical, or it could be from electricity that you obtain from the sun. So photovoltaics might be an efficient way of getting the full spectrum rather than just the red and the blue. And there'll be various special cases for applying this to deserts, applying some of the agricultural concepts to carbon sequestration in the Arctic, and then for providing food in sealed environments such as you might need for colonies in space.
You mentioned sun as a source of energy, and so this might be a good time to move to a question that Beth B just asked, which is, can SynBio be used to manufacture fuels cost efficiently? Also, can SynBio be used to affect climate change by sequestering CO2? And I know you've been involved kind of in this over the years. I think it might be a good opportunity to also speak to some of the companies that you've had kind of in this space. And then there was a follow-up question about companies you've had in the SynBio food space as well.
Right. So like I say, foods and fuels are tough, difficult, because there's already solutions out there, and they're very inexpensive, cost-effective solutions. We can make greener solutions, meaning more eco-friendly solutions, but even that's challenging to do that cost-effectively. I think where we can really succeed is in carbon sequestration and prevention of carbon escape. In the Arctic, we have 1,400 gigatons of carbon, which is like 200 times more than the total human production per year. And by the way, even the most exotic strategies for reducing human productions, there still is the accumulation. That if we produce any carbon, that tends to accumulate, and we need to reverse it. So anyway, the synthetic biology, I think, can make nearly free, after a small initial investment, nearly free ongoing carbon sequestration and trapping of 1,400 gigatons of methane in the Arctic, plus new photosynthesis. I think that's a more interesting way of thinking about it than in terms of biofuels. I think that photovoltaics is getting more and more efficient. It doesn't self-assemble the way that forests and farmlands do for the most part, certainly forests. But it does have advantages for cities and farms that supply them.
And can you speak to some of the companies that you've had kind of in the agriculture, food, and energy space?
Yeah, not so many, partly because it's so hard to go beyond where we are. Some are aimed at, so we have work with Memphis Meats and some other groups that are interested in catering to people's tastes. I think the idea of developing better tasting food has a lot of promise, and taste and texture. I had two companies, Jewel and LS9, that were acquired by companies that produce a lot of carbon dioxide, so that they can immediately go in from the carbon dioxide production to sequestration. I'm not actively working with them right now, so I don't exactly know where that stands, but there were two acquisitions. but I think we have an even bigger play in the Arctic. Both the Arctic Ocean and the landmass are far more productive than the rest of the world, which a lot of people find surprising because it seems so cold that there wouldn't be much action. But there's high nutrient content in the Arctic oceans and high sequestration in the Arctic land, where you've got these 1,400 gigatons of methane because you've got 500 meters, some places, of topsoil relative to, say, 1 meter in say, the Amazon rainforest. So a lot more carbon at stake in the Arctic.
Super interesting. I like Manuel Jose Leon's question a lot. When I spoke with Ginkgo executives, they have this vision. Ginkgo Bioworks executives, they have this vision that SynBio can be a $90 trillion industry. In other words, touching almost every fabric of life. But many have reservations about whether it's going to be able to impact iron, steel, kind of those natural or synthetic metals. And so Manuel's question is, how is SynBio going to change the metals and mining industry?
Yeah, I'm surprised that my friends at Ginkgo were so restrained in saying it will only affect living systems, because I think they know, as I do, that biological systems make inorganic systems quite well. They make something that's very close to a perfect fiber optics in sponges. They make ferromagnetic materials. They make reduced metals, which is getting close to your, and they do it with atomic precision. So now it is true that many of our materials don't require atomic precision, but usually it doesn't hurt. If you think of some of the finest steels that gave nice swords and so forth, those were actually quite exotic at the nano realm. They just were made by cruder methods. Anyway, I think that we're just warming up synthetic biology for inroads into literally every kind of manufacturing. I don't think there's a chemical bond that we can't make biologically that I know of.
Cool. I love it. I love it. I love that. We've got questions that are in the realm of machine learning and quantum computing. So folks are wondering, what's the role of machine learning in SynBio? Is it critical? Is it not so much? When is quantum computing going to come to SynBio? The first question, Mariana Milhausova, again, asked in chat, but there have been a number of quantum computing and machine learning questions in the pre-submitted questions as well.
Yeah, I think the quantum computing field is missing an opportunity. Again, they're restricting their views so that they're only solving certain kinds of problems that are well formulated. Encryption, decryption problems, things, some of which are inherited from the Silicon von Neumann era, just now considerably more efficient. But I think what's being missed is the possibility of natural computing. So there's a Wikipedia page on natural computing where you can, for certain kinds of computing, making lots of different instances showing a diversity of solutions can be more cost effective than simulating them. So trying to simulate, let's say, a quantum structure of a protein is far less efficient than making the protein itself, or making millions of different proteins and seeing and observing what their functionality is. Trying to simulate that with a quantum computer would be extraordinarily inefficient. And there's just the number of examples of natural computing is vast. It's just it's not pure enough, I guess, or some it's too obvious or it's too unobvious. I don't know what it is, but I think there's some great opportunities there for natural computing, which is essentially synthetic biology.
Great. One question that's super that very much resonates with me, so I want to make sure we have a chance to ask it. is voices from outside the traditional biotech community are describing a shift in the ecosystem that will allow more and younger entrepreneurs to start biotech companies. What is your perspective on this? Boston's a unique ecosystem for biotech. Do you think other regions will be able to catch up? And what does it take for an ecosystem to become successful? Do you think there will be biotech founders without graduate training, just like in tech today?
Yeah, I don't think. I think there's room for a lot of diversity of thought. I think that outsiders are often welcome. It helps if they pair themselves with people who have deep domain knowledge, and the deep domain knowledge does probably require graduate work. But you can have teams that are hybrids of various sorts. I think it is hard to develop the kind of concentrated teams that occur in the Boston area. Even on the West Coast, there's such an emphasis, such a success story with electronics, telecommunications, and internet, that it's very distracting. It's faster payoff, perceived payoff, than in biotech. But in Boston, you have this concentration, and the concentration is a positive feedback loop where it gets more and more, it attracts more and more of the best and brightest in that field. And I don't think that's harmful. I think that having concentrated centers of excellence, each country, each city can have its own thing, but and there's some reason to distribute it as well. So now we're in the virtual world, and more companies will, when they hire an individual, they won't relocate them. In which case, you can have a center of excellence with spokes that go out all over the world. So I think the whole world will benefit from both the virtual reality that we're in, partly due to COVID, and to these hubs and spokes models.
So we've only got literally 1 minute left, and I'm sensitive that you may have a hard stop at 2. So between CRISPR, predictions, your personal productivity, your book Regenesis, what are you most in the mood to field?
Most what?
What are you in the mood most excited to field? I've got questions on more predictions on CRISPR, on your productivity, on Regenesis. What question would be exciting for you as we're running out of time?
I'll do all three of them quickly. I'll run a tiny bit over.
Okay.
So I don't think CRISPR is special, just like I don't think the very first next generation sequencing method we worked on was that special. There are going to be multiple solutions. We're already seeing that one of the best players in CRISPR is not the CRISPR part; it's the deaminase part for base editing. But we also have transposinases and integrases that companies like Tessera are using, which have nothing to do with CRISPR. In fact, they have the advantage that they're derived from human proteins, so they're not subject to immune rejection. So I think that the great breakthrough in CRISPR will be abandoning CRISPR. Maybe, at least in part. The same thing is that, the greatest breakthrough in previous generation sequencing was switching to next generation sequencing. Sequencing is dead, long leaf sequencing.
This is a perfect time for the question, which was, what qualities do you think the technology that replaces CRISPR should have?
Well, I think it should be efficient, easily delivered. For certain purposes, it should not invoke an immune response. Being multiplexable, so that the goal is not to get 80% editing, is to get 100% editing at 100 sites or more. So there's lots of room for improvement. But we'll have a little of everything. There's still people that are using programming languages that were invented in the 60s. So we shouldn't be in a big rush to dump the older technologies. Regenesis, the book, I am writing a new book. It will be quite different, but hopefully we will have learned how to do an even better job in the intervening 6 years. So.
The question for Regenesis was, will you be updating it? And are there other books on SynBio that would get folks up to speed on the current state of the art, like Regenesis did when it was released?
I wish there were. The book that I'm writing is not exactly an update, but it will be even, hopefully, even more mind-boggling in its own way. That's all I can say about it right now.
Perfect. And then David Roth asked, what is your production function? In other words, how do you get so much done? What's the secret formula for all the things you've accomplished?
Well, I think the first thing is everybody's being very nice to me and saying that I'm productive. You know, we work. I mean, in fact, we try to be unproductive in that we're trying to be disruptive, out of the box sort of thing, and that means a lot of those are going to fail. We just want to learn from those failures as quickly as possible and recombine things. So creating an environment where people can follow their dreams and learn from successes and failures very quickly to recombine multiple methods. So there are not that many environments where you have so many different things going on at once that gives you a toolbox to draw from.
Awesome. We have many more questions, but we won't be able to get through all of them. George, thank you so much for your time and for going over a little bit. We're really grateful for it. I don't know if you have any closing words or anything like that, or we could just end it here.
I don't really have time for closing words because my next will be waiting, but I really enjoyed this, and I'd be happy to do this again if you have more questions. So, and good luck to everybody that's on the call. Thank you for being there and participating in this revolution.